Handsome ASMR, back with another video. Today we're going to be talking about the central dogma of molecular biology. What is a central dogma? It is what describes the flow of genetic information within a cell. And the oversimplified principle behind the central dogma is that DNA is replicated and then eventually transcribed into your RNA, and then your RNA is translated into your protein. Overall, this process underpins all cellular function and, importantly for medicine, many diseases can arise from errors at one or more of these steps. Let's talk about the first step, which is DNA replication. The purpose of this step is to ensure that each daughter cell receives an identical copy of the genome. This is clinically relevant because mutations during replication can lead to cancer. For example, the BRCA1-2 gene mutation is known to be associated with the development of breast cancer. The second step involves transcription, where we convert DNA into your RNA. Transcription happens in the nucleus of the cell, and it is facilitated by the RNA polymerase. So the RNA polymerase is the enzyme that does the transcription. Your final product should be the mRNA, which stands for messenger RNA. This step is also clinically relevant because defects in transcription factors can play a role in the development of various malignancies or cancer. And one classic protein that is a transcription factor is your p53. The final, or this third step, is RNA processing. This special step occurs only in the eukaryotes, and it consists of the following events. It involves capping the RNA product at the 5' end, and then the polyadenylation of the RNA at the 3' end, as seen over here. So here's a 5' end, and here's a 3' end. This occurs after the introns are spliced, or in other words, the introns are removed. This is how you get the final product. This step is also clinically relevant because splicing errors can cause diseases like beta thalassemia or spinal muscular atrophy. Beta thalassemia has to do with some malfunction or deformed structure of the red blood cell. I can talk about this more in future video if needed. Alright, the fourth step is translation, where RNA is translated into your protein product. This time, this step occurs in the cytoplasm, not the nucleus. Specifically occurs at the ribosomes in the cell. The key players in the translation process is your mRNA that you produce through transcription, and then your tRNA that helps to bring each individual amino acids to build an amino acid chains, and of course your ribosome, which is where the actual synthesis of the protein or the amino acid chains take place. This step is clinically relevant because antibiotics like tetracyclines and aminoglycosides target bacterial ribosomes specifically. So what happens after you build your initial protein product through translation? Well, now you have protein folding and possibly some post-translational modifications. This is important because it helps to synthesize the final protein product with a unique structure. This final structure will ultimately determine the protein is the FTR protein misfolding. So other clinical concepts that involve the central dogma the central dogma include DNA mutation. And so one example of this is your sickle cell anemia, where the glutamic acid residue is substituted for the valine residue. There's also the splicing defect that can occur, and this is, or one of the pathologies that can result from this is spinal muscular atrophy. You also have misfolded protein. This can once again lead to Alzheimer's. It can also lead to prion disease. And then you have your transcription factor mutation, which can lead to Liv-Promeny syndrome, involving the p53 protein. So in summary, here is a nice simplified diagram describing the central dogma. You have DNA that can be replicated to form more DNA. Alternatively, DNA can be transcribed to produce your pre-mRNA product. And then the pre-mRNA can be processed into your mature mRNA, which has only exons, and all the introns have been spliced. And then your mature mRNA can be translated into your protein product, whereby the protein can then carry out its function. So before I end this video, I just want to draw your attention to this helpful picture showing the different forms of post-translational modifications after you create your initial protein product. These steps can help contribute to the protein's unique structure and ultimately determine its function. So that is it for today's video. Like and subscribe for more ASMR content like this, and I'll see you next time.